today's seminar speaker, Dan Reuman. Dr. Reuman is professor at the University of Kansas in the Department of Ecology and Evolution Biology, and a senior scientist at the Kansas Biological Survey, and a James S. McDonald Foundation scholar. Dr. Reuman holds a BS in mathematics from Harvard and a MS and PhD in mathematics from the University of Chicago. And while his initial background is in pure math, he has built an impressive research program applying mathematics to understand complex ecological questions, including metapopulation dynamics and population synchrony. His work on synchrony has long fascinated us in our lab. He has conducted foundational work on how synchrony amongst populations is structured across landscapes and multiple timescales, and helped us look at housing systems in a new way. He is an ideal collaborator, able to engage with empiricists to identify and powerfully test new questions in population and community ecology. Personally, I had a great opportunity to be mentored by Dan as an undergrad at the University of Kansas, and he has already contributed to my ability as a scientist today, inspiring me to continue my work in species programs. Oh, sorry. Let's hear it for Dan. Take it away.
Thank you very much, Jasmine. Can everybody, I don't know where these different mics go, but can you hear me okay if you're here? All right. So I'm going to talk about the title, The Importance of Asymmetric Relationships. If I can, there we go. This talk is not about this. We're not talking about relationships between humans. So let me say what we are talking about. It's a data kind of talk, but I don't want anyone to get bored about like, oh, this is going to be a talk about statistics. I guess it is. And anyway, that shouldn't bore you. But I hope to convince you by the end of this to take a look at whether you see this in your own data. Because I think it is something that is really quite simple, really quite broadly applicable from what we've tried so far, but doesn't get taken into account much in ecology. And we've tried to see the importance of this phenomenon in several different concepts in ecology, and we continually find that it does play a role. So that's my kind of agenda. So let me start by defining what this thing is. So we call it asymmetric tail association. Let's just look at a couple scenarios here. These are made up data just to illustrate the point. We're imagining it could be populations, it could be other data sets too, but for this example, let's just imagine that you've got two different populations in two different places tracked through time, like measured every year, let's say. And so I've written down two time series here, but then let's make a plot on the right, which is just a scatter plot where one axis is population 1 and the other axis is population 2. And I've got two scenarios here, scenario 1 and scenario 2 in the bottom. And both of them, I've engineered these data to have a Pearson correlation of 0.65. But you can look at these two scatter plots and see a difference. And that's what I mean by asymmetric tail association. So in this case, they're more tightly related to each other down here in the left portions of the two distributions and less tightly related up in the right portions of the distributions. And here it's the reverse. Now, the effect isn't strong in this case. Sometimes it is strong. Sometimes it's like this. And what difference does it make is what we're going to explore. So we've just called this left tail, we'll say left tail association dominates in this case. We'll say right tail association dominates in this case. I just want to point out that we're kind of relying on there being a positive association between the two variables if we want to use the language of left tail or right tail association. If there's a negative association, we'd be talking about stronger association in the left tail of one distribution with the right tail of the other. But that's like a detail that we'll just sweep it under the rug for now and be thinking about positively associated variables. So ATAs is the abbreviation I'll just consistently use for asymmetric tail associations. So practically, what does this mean for populations? Like if we can think about other types of data sets, but I like populations. So let's just to put some like reality on this, let's talk about what it means for populations. If you have stronger left tail associations between the dynamics of two populations, then that means that when they're low, when they crash, they tend to crash together more so than they tend to explode to high values together. versus if they have stronger right tail association, it's the reverse. It means that the outbreaks tend to happen more together than the crashes. And you can imagine if you're an ecologist and you're interested in something like rescue effects, that'll make a big difference. If the populations tend to crash together, that's going to make rescue effects harder than it would if like they didn't crash together, but they outbreak together. On the other hand, if you're interested in pest control or disease dynamics, then it matters whether they outbreak together. You don't want them to outbreak together. You're okay with them crashing together. So we can start to see some of the reason for this. Our first paper that we wrote on this was a big part of it was like a survey. We wanted to look at a lot of different data sets in ecology and see if we could see this asymmetries, asymmetric tail associations in the different data sets. And we find that you can a lot. So here are some examples, which I'll do a little more detail later, but soil nitrogen and carbon content from a bunch of different locations, basal metabolic rate and body mass of birds and mammals. And I've plotted the data as in their sort of native state on the top here. And then I've plotted a ranked version of the data on the bottom. It's a normalized rank. And so I just want to point out things like, look at this. It's only in the very most highest values of carbon and nitrogen where those two values become like super tightly related to each other compared to the rest of the distributions. So this is a kind of asymmetry here. You can see a little bit of asymmetry for reasons that I don't understand. Basal metabolic rate and body mass seems to be more closely related to each other when those two numbers take big values than when they take small values. So asymmetric tail associations are not accounted for by standard measures. And to do that, here's some more manufactured data. So I've got multiple panels here, but the ones on the left and the ones on the right were always engineered to have the same Pearson correlation, that's the P, and also the same Spearman correlation, but obviously not the same, right? So this and this both have Pearson correlation 0.87, Spearman correlation 0.9, but this is left tail associated, this is right tail associated. If this was like, you know, measles outbreaks in New York and Chicago, this would be practically quite different from this. Or if this was like aphid dynamics on two farms, it'd be a difference which way, well, like which of these situations you were in, even though standard techniques don't take account of that. So we've done several papers now exploring ATAs and documenting ATAs in ecology and looking what difference it makes for, you know, phenomena in ecology that people care about. And it turns out they're important for lots of different phenomena. So my goal is to summarize some of that work, giving an emphasis on ideas and minimizing technical content and sort of hoping to lead you into thinking about ATAs. Before I get started on the ecology, I wanted to give like a taster idea, unrelated to ecology, but super cool, I think, which also illustrates kind of where these ideas came from to some extent. They come from the statistical literature. They also come from the financial literature. And also just, it's a good example. So back before 2008, people used to bundle subprime mortgages. So this was the whole 2008 financial crisis. People would take a bunch of mortgages of people whose credit rating might not be so good, and they would claim that by bundling them all together, that was a safe investment product, because even though those people might individually have a high default risk, they wouldn't all default together. So they were supposedly uncorrelated or limited correlation, right? So the idea was if the default risk of different mortgages all fluctuated and it was uncorrelated, these default risks were all uncorrelated with each other, then when you aggregate the whole thing up, you get something nice and stable through time, right? What they didn't take into account is that they estimated correlations between default risks using short time series, like normal times, like the last 10 years, right? If you're using a short time series, you're almost by definition, not taking account of the tails of the distributions, because it's a short time series, you haven't explored the tails, right? So they didn't take into account the fact that the correlations between these default risks might be different in the tails from what it was in the center of the distribution. And what you actually have is that when you have extreme times, everyone's default risks are quite correlated. which is kind of obvious if you think about it in hindsight, because like if the whole economy goes south, then everybody at once is going to have trouble paying their mortgage. And then they all become correlated. And the investment product, which was the bundle of subprime mortgages, has not just, it no longer has this nice stable property, it has this crazy risk of default. And that's basically the 2008 financial crisis in like a super short nutshell. And in fact, you can find papers that attribute it to this and attribute it to one particular modeling philosophy that didn't account for tail association that lots of people happen to be using. So I just want to point out this is housing defaults. Maybe you don't care about it if you're an ecologist or an evolutionary biologist, but there's no reason why these same ideas wouldn't apply. Like if you're imagining different species together, maybe uncorrelated, different plant species in a pasture or different tree species in a forest. But if some bad time comes along, like climate change or something, that has a decent chance of affecting them all in the same way, and then they become correlated and we have the same kind of problem. This is what I said, so I'll skip it. Okay, so here's an outline. I'll give some examples. I'll give some of the mechanisms that we've shown produce ATAs in ecological data and one evolutionary case. And then I'll give some of the consequences for ideas of ecological stability and coexistence. Start with some examples. So the first question one of the first questions we wanted to look into was just how common are these things? And we started, since I do a lot of population dynamics, we started with population data of, let's say, the same species in different locations. So we had data from the Roth-Hampstead insect survey on the abundance in different places around the UK of different species of aphids. So this is an example of one of these species of aphid, the green spruce aphid. There were 10 locations across the UK, long time series from each locations. They have these suction traps that are basically like a giant upward pointing vacuum cleaner that's always sucking. And anytime an aphid flies by, it gets sucked in, and then they have people who count the samples. And they've been doing that for 40 years-ish. So it turned out when we looked at ATAs between different pairs of locations in this time series of green spruce aphid abundance, 95% of those pairs showed stronger left tail association, like more correlated crashes than outbursts, compared to right tail association. So that's an example. Here's an example from the ocean. So this is from another remarkably similar long-term monitoring effort, the continuous plankton recorder. They have a room of people that count aphids in the Roth-Hampstead insect survey. They have a room of people that count plankton in the continuous plankton recorder survey. They gather the data in different ways, but you end up with time series once you've processed the data. So we had data for this particular phytoplankton species, Serratium furca, from 14 areas in the North Sea. And when we again looked at pairs of time series from different locations, and 79% of pairs showed stronger right tail association. So the opposite from the aphids, but we still had this quite strong bias towards one of them. I don't know, in this case, I don't know why it's left tail for aphids and right tail for Serratium furca, but our goal here was just to see, like, do you see this? And it turns out we did a lot of other examples. ATAs are common in spatial synchrony. So if you measure like the same species in different places, get those two time series, look for ATAs, you frequently find them. And we've also shown, if you have multiple different species in the same place, you often get ATAs between those too. So we did examples, that Roth-Hampstead insect survey example, they don't just calculate the abundance of green spruce aphids, they do all the aphids that fly into their trap. So we were also able to look at different species of aphids captured by the same trap, and you see ATAs then too. Here's an example of phenology. So in addition to the abundance, this Roth-Hampstead insect survey, they capture the aphids and they count them daily, so you can get phenology information. So we processed the data to get aphid first flight time series for 20 species at one particular place in the UK. And we put them into a matrix like this. The red over in this matrix means stronger left tail associations between that pair of species. The blue means stronger right tail association. Then yellow was just they weren't correlated at all. But you can see more blue than red here. So what that means is species phenology in this example, in this location, is of the different species more strongly related when late. So when it's a late year for the aphids, when they fly later, it's more, the different species tend to be more related, more strongly related. So going back to these examples I did before, this is soil N versus C at lots of locations in the UK, in the US. And this middle one is metabolic rate versus body mass for mammals. Each dot is a species here. Each dot here is a soil sample from a different place. And this is the same, but for birds. And each dot is, again, a species of bird. So there must be, of course, different mechanisms, since we've got like soil pits on one hand and species of birds and mammals on the other hand. So we're going to explore some of that. First, I want to say how ATAs are measured. Since I promised not to get too technical, I won't, but I do want to at least like give the idea of a basic measurement that we cooked up that will do part of the job of measuring these ATAs. There are lots of ways of measuring it. It links into a well-established field of statistics called, where you study a mathematical object called a copula, but I just want to give this basic idea. If you want more on the technical side, I'll give you references at the end, and I'm happy to talk about it. So if you take your data and you rank them, just as you would if you were doing a Spearman correlation, and so we'll use a normalized rank where you just rescale all the ranks so they fall between 0 and 1. And then we're going to calculate this, which looks like the Spearman correlation, except we're only going to compute the sum over the points in the portion of the distribution that you're interested in. So like if you're interested in left tail associations in the entire left half of the distributions, you might just sum over these points that are white here. Or in this example, sum over these points which are white. And then you can contrast that with computing the same thing, but taking the sum over the other points. And so one will give you a measure of left tail association, one will give you a measure of right tail association, and you can compare those things or take a difference or something to measure asymmetry of tail association. And this is, a lot of times when the data are not super abundant, you just want to split the data in half and do left versus right. If you split, you can look at like the bottom 10% and the top 10%, and that's fine, but you're going to need more data to do it. Okay, so let's think about causes of why we would see this. So I'm going to sort of do, a lot of times I find that I like to start from the empirical situation. And a lot of times we find by analyzing data, we later come to some kind of conceptual or theoretical understanding, but it emerges from the data. And that's what happened in this case too. So I'm going to present it starting with the idea, almost as though I came up with this idea just out of thin air and then it happened to play out in the data, but that's not how it happened at all. We started by analyzing the data and then we figured this out afterwards. But it sort of makes a better presentation to start with the data. Start with the idea. Okay, so let's imagine a situation where there's lots of populations in different locations and they're all influenced in some sigmoidal way by some environmental driver. It's like a totally normal situation, right? The example that we're going to imagine is kelp forests off the coast of California, and they get affected by waves. If the waves are strong, it rips up the kelp. If the waves are weak, it doesn't affect the kelp. If the waves are really strong, it just rips them all out. And so making them twice as strong as that has no further effect, right? But if they're in between, the strength of the wave is going to make a big difference, like how much of the kelp gets ripped out. So that's why it's sigmoidal. Like the difference between really weak waves and weak waves is basically nothing. The difference between really strong waves and strong waves is basically nothing, but in between there's this threshold-like behavior. And then we're also going to imagine that all the populations have some local independent effects influencing them that aren't related to waves. And now we're going to imagine two regions, one of which the waves from year to year fluctuate somewhere in this range, like you can imagine a sort of a normal distribution that spans about this range. And then in the other region, the waves are on average stronger, and they fluctuate kind of in this range. And if you've got those two scenarios, what's effect is it going to produce? Well, down here, when you have, you're going to get more right tail association, because only when the waves get into their right tails does it really have much of an influence on the population. And if the waves are spatially synchronous, then it'll produce synchrony, but only in those right tails. And so just by simulating a setup like this, what you end up with is right tail associations between the population and the environmental driver, and also right tail associations between two populations which are influenced in that way. Versus if you're up here, where the waves are on average stronger, then where it really has a differential effect, depending on the strength of the waves, is when the waves are on their weaker end, right? Because when they're on their higher end, it's just ripping out all the kelp. And it has the same effect regardless of how the precise value. And so you end up with the left tail associations up here. And we checked this. I mean, having filled in the idea, I don't necessarily need to go into the great details. You can read the paper. But basically, that's what we found for kelp. There's amazing data from kelp from my colleagues Tom Bell and Kyle Cavanaugh. They have satellite information where they can detect the kelp when it reaches the surface, super high resolution, going back 35 years or something. So that's one mechanism. And that's one that I kind of think probably is somewhat general, like the ingredients that went into that explanation, none of them was like shocking or unusual. Like you might expect that kind of sigmoidal effects to happen to lots of different environmental variables, lots of different systems. So you might find this in other systems. I want to do one that's evolutionary, even though we've mainly done ecological work on this stuff. And this is really just partial. So we've really kind of just scratched the surface on the evolutionary side of it. But having seen some ATAs in the body mass versus basal metabolic rate for birds and mammals, we felt like we needed to at least attempt some kind of explanation. And it's only an explanation like, you know, we give an explanation which is driven by something, but we can't explain why that thing might happen. So we're just kind of kicking the can down the road. Okay, so we showed by simulation that if you have a phylogeny and you imagine the evolution of two characters down the phylogeny, and you imagine that evolutionary increments, as you move down the phylogeny, have ATAs, like the increments themselves are correlated more in the right or left tails, then what that translates to is if you look at the values of those characters across the extant species at the tips of the phylogeny, you'll also get ATAs there. So if you're willing to assume ATAs in the evolutionary increments, you get ATAs in the extant characters. Now, what we can't, I have no idea why you'd expect to have ATAs or not in the evolutionary increments. So we kicked the can down the road. If someone has ideas about that, I'd love to hear it. Okay. There's some additional causes which I'll just mention. We've also showed that if you've got ATAs between environmental variables in two locations, then you're going to end up with ATAs in the populations that they influence in kind of a Moran effect, like an extension of the Moran effect. And probably there's other causes of ATAs that we haven't thought of. So let's say something about consequences. So far I've documented that, or I've argued that these are common. I've given a couple reasons for why they should be found, but I haven't talked about like why you should care. And that's where the consequence part is. So I want to talk about ATAs and their influence on ideas about ecological stability. And I've already kind of alluded to this, but let's talk about it in metapopulations. If populations in a region tend to be right tail associated with each other, then they tend to outbreak together more than they tend to crash together, which means if you're interested in, let's say, the total population across the whole area, like the spatial total population and how it fluctuates, or the spatial average population and how it fluctuates, you're going to find spikes at the high end, because that's when everybody goes together. They go together when they go high, but you're not going to find spikes at the low end. So I could call that unstable on the high end. Like one thing that I find, like well, the stability literature, oftentimes, depending on what you define by stability, and there's a whole arguments about that and so on. But a lot of times people talk about just how stable a population or a set of populations or community. But it turns out you can have stability at the high end is different from stability at the low end. There's different kinds of stability even within all that. On the other hand, if a population in a region, if all the populations in a region tend to be left tail associated, they crash together, but they don't outbreak together. And when you take the spatial average or the spatial total population, it's going to have big downward spikes, but not big upward spikes. So then the regional total biomass or the regional total population will be unstable on the low end. And might also be prone to regional extinction. And I'll come back to an example like that in a minute. So in addition, the kelp example, we extended that a little bit further. It turns out that with the actual kelp, because we had this really, the data go all the way down California and down into Baja California. In regions like Region 1 that had this kind of influence, total regional populations will tend to have bigger booms and busts, and we did see that. And in regions like Region 2, the regional total population will tend to have bigger busts than booms. The same, if you're a community ecologist instead of like a spatial ecologist, then you might be interested, instead of looking at different populations in different places of the same species, you might be interested in lots of populations of different species in the same place, right? That's a community. So you're interested in community dynamics, and the same kind of idea applies. Now the time series are abundances of distinct species in the same place, and the aggregate time series might be the total abundance of the community or the total biomass of the whole community. Like maybe you're a plant ecologist and you're interested in lots of different plants in the same quadrat, and they all fluctuate, and sometimes they're correlated with each other, and sometimes they're negatively correlated with each other. And you might be interested in the fluctuations of the total biomass, plant biomass in that quadrat through time. So how do ATAs between the species influence stability of the aggregate? And we explore this theoretically and empirically in this paper. I want to start by giving a theoretical example, because I think it helps illustrate the idea. And then we have actually a couple of empirical examples. So this is made up data again, and it's 20 species. And I've broken them into red and black groups, which don't mean anything except it's just two groupings. In this scenario, you can see by looking at the data, you can see that the black tends to be high when the red is low and vice versa. But you can also see that whenever any of this, either color is high, the lines become disaggregated. And whenever they're low, they're all together. So this is a situation of left tail association. So we've got two compensatory groups that tend to cancel each other out in their fluctuations. But within each group, when they're low, they're all doing the same thing. And when they're high, they're doing somewhat different things. And you can imagine this kind of case, like let's say there's a drought kind of variable that's at play. And the species, when water is scarce enough, they're all going to get hammered and they're all going to be correlated. But when there's enough water past some threshold, other things are going to control which one is more abundant. Like there's enough water basically for them all, or for most of them, and there's something else influencing them differently. So they compete on different things at that point. So if this is the case, and then all this time series is just the sum of all these time series. So that represents, let's say, the total biomass of all the plant species. And you can see these crashes are exceeding this tolerance line right here. So if you're really interested, if the disaster situation was going below this dashed line, then you're going to do it more than you would if you didn't have this structure over here. And this is just the distribution that you get. And you can see the left tail goes below this dashed line more than the other example, and than it would if it weren't for this tail association. And here's the reverse case where I've just flipped everything. So now they're correlated when they're high. Now they outbreak together. And now you can see booms exceeding the symmetric threshold on the opposite side of the distribution. And now you can see the right tail going up here. And you don't need, like, the difference between, a probability of exceeding a low threshold of 4% in this case versus like 0.1% in this case, if it's a real disaster, then that's big. If like that corresponds to something that you really want to avoid, then that's a big deal. So we did take some real data and analyze it from this perspective. We had two long-term community data sets, one from Hays, Kansas, a long-term grassland monitoring project, and another from Konza Prairie, Kansas. And species associations happened to be mainly left-tailed associated, the different species in the quadrats, mainly left-tail associated at Hays. They happen to be mainly right-tail associated at Konza, and it worked out as the theory predicts. We have methods like bootstrapping type randomization type methods where you can like rearrange the tail association. And when you do that, you can find that for Hays, where there was mostly left tail association, it meant that community biomass didn't crash as low as it otherwise would. Sorry, Hays was right tail associated. So community biomass didn't crash as low as it otherwise would if it weren't for that. And Konza was the reverse. So I guess the lessons from this part of it is species dynamics can be more or less related to each other in one tail compared to the other. Two species might appear to fluctuate independently in normal times, but might become correlated in extreme times, which it's a little bit scary in the sense that with the normal data set, you can't necessarily assess what's going to happen with aggregates when extreme times come because you just haven't sampled the tails. And we're headed into times where we might expect to be sampling the tails more than we have in the past. This is a theoretical result. ATAs, it's hard to study extinction risk except for if you do it theoretically. So we did it theoretically. Synchrony is known. Synchrony between different areas, like if you've got two populations which might exchange some migrants, the extent to which they're correlated to each other influences the overall risk of the whole metapopulation going extinct. That's well known. But furthermore, whether that synchrony has left or right ATAs in it is further going to influence the overall extinction risk. And so I want to just illustrate that right here. So if your two populations are under left tail association, then that means these, what we call bonanzas in this example, don't happen simultaneously, but the catastrophes, the big crashes of the two populations do happen together, which means that rescue effects are harder. Like if one population is low and needs to be rescued from migrants from the other one, well, the other one is more likely to also be low and therefore contributing fewer migrants. And so this would tend to elevate the metapopulation extinction risk if you have this left tail association. But on the other hand, if you have right tail association, the catastrophes don't happen together. Instead, the bonanzas happen together. And then rescue effects can happen like normal. In fact, better than normal because if you've got the same amount of overall correlation, but now it's allocated to the bonanzas, then that means you have less correlation in the catastrophes. And then I want to say something about work that I did with Jasmine, actually, when she was at KU before she came here, about applying these ideas to modern coexistence theory. And this will take a little bit of warm up, which I want, because modern coexistence theory, it's like well-known important theory, but it's also kind of hard and some people don't know much about it. I didn't know much about it until we started this work. So I want to give a little bit of background before saying what ATAs have to do with it. So modern coexistence theory, in a nutshell, breaks species coexistence down into the contributions of different mechanisms, which have names. One of the names is storage effects, and I'll explain something about that in a minute. You've probably heard of that. And that's the one we'll be focusing on. So storage effects hinge on associations between competitive pressures, which I'm labeling as C, experienced by a currently rare species, and environmental boosts that it might get, like how good the environment is for it right now. So modern coexistence theory, what it kind of focuses on is the ability for a species when it happens to become rare, can it recover? Like that's the idea of coexistence. If all the species, when they happen to get rare, can recover, then you can have coexistence. And so it wants to attribute the ability for these species to recover when rare to different mechanisms. And one of them is storage effects. So let's delve into that a little bit more. So the C is when you've got one of your species rare, it's experiencing some competitive pressures, and it's got an environment that's affecting it in a certain way. So we're going to label that environment by E. So for the currently abundant species, good environmental conditions, which is high E, tend to be paired with strong competition that it's facing because it's abundant. So most of the competition it's facing is intraspecific competition. It's coming from members of its own species because it's abundant, right? So if it's a high E year, like a really good environmental year, it's also going to tend to be a heavy competition year, a difficult year for competition, because it's the abundant species. But on the other hand, if you're a scarce species right now, you're currently a rare species, then those two are going to be less coupled because most of the competition that the scarce species is experiencing is from the other species because it's rare. So it doesn't exhibit much competition on itself. Instead, it's getting whatever competition it's coming from the other species. So that tends to uncouple those two things. And a good environmental year for the scarce species is not as related to high competition for that species. And that's the asymmetry that the storage effect mechanism is capturing, like that gives an advantage to the scarce species. And that's the key of the mechanism, like the fact that it applies preferentially, it gives the boost to the scarce species. And then when they eventually switch, if you end up with the other species becoming scarce and the first one becoming common, now the advantage goes to the other species, which is now scarce. So this asymmetry is what gives a benefit to whichever species is rare. Now, the thing to notice here is that the storage effect, this mechanism, hinge on associations between E and C and the strength of those associations and the nature of those associations. So the question that we wanted to ask was, what about ATAs? Like, what if you're in a situation where E and C are left the same overall correlation, but left tail associated versus right tail associated? Then is that going to make a difference? So in a nutshell, what we did, like the classic MCT has been around for a while, and it takes a quantitative measure of coexistence of these species, and it decomposes it into these mechanisms. So you can say, like, how much does storage effect contribute to coexistence? And how much do these other mechanisms, which I'm not going to talk about, contribute? All we did, well, I shouldn't say all, what Jasmine did, which was great work, was take this storage effect and break it up into ATA effects versus the rest, which we'll just call EC correlation per se, but maybe the rest is simpler. So it's a further decomposition. So we extended the modern coexistence theory, splitting storage effects into the ATA effects and the rest. We have some arguments that ATAs aren't going to influence these other mechanisms because the only mechanism that has to do with correlations at all between E and C is storage effects, and ATAs are about correlations and relationships between variables. So we used models and we used a laboratory diatom system by Descamps, Julien, and Gonzalez to then apply this theory, this new decomposition, to some real systems, because we wanted to find out, like, does this term matter? A priori, it could be that this term is always vanishingly small, and it just doesn't matter for real systems, and we might as well forget about it. Or it could be that storage effects is 90% this and 10% this, and we wanted to know which situation we were in for some real systems. Well, real and real-ish systems. So ATA contributions to coexistence, it turns out, across the models that we looked at and the one empirical system that we looked at, it's hard to get empirical systems where you have enough of the technology to do this with. It turns out that ATA contributions to coexistence are often, but not always, of a similar magnitude to the other mechanisms, which basically told us that it's worth thinking about. And then we were able to show that ATAs can actually make the difference between coexistence and competitive exclusion. So you can find cases where your two species coexist, But if you took out ATAs, if you like did your bootstrapping so that ATAs were no longer present, then one would outcompete the other and you'd lose one of the species. And you can also have the vice versa case where two species don't coexist, one of them outcompetes the other, but if you took out ATAs, then they would coexist. If you want to know more about that, you've got somebody here who you can talk to about it any time. So I just want to give a summary, and then I think there should be like plenty of time for questions and discussion. I hope people have things they want to say. Summary is that ATAs between ecological variables of lots of types are common. And there's some reasons why they can arise. One of them is nonlinear effects of environmental variables, which is something that should be pretty common. There's also, by the way, no reason to expect symmetric associations. Like if you, in some sense, having two variables randomly chosen, whatever that means, be symmetrically associated is like if I ask you to choose a number and you choose 0. Like the chances of that are 0. Most likely, if it's sort of randomly distributed somehow, we're going to get some variables which are right tail associated and some that are left tail associated. And the case of symmetric association is a special case. It's really a measure 0 set in some space of distributions. So nevertheless, ATAs are seldom considered. I'm not blaming anyone. It's like it makes sense that when you start out studying a problem, you use a simple metric like correlation. And probably that's the thing you mostly want to use. But I think it's worth thinking about after you've got done thinking about what you can get from correlation. Is it worth taking the next step and thinking about ATAs with your data? And then we've shown that they influence ecological stability, species coexistence. There's some other phenomena. We have some results about Taylor's law. If you're interested in Taylor's law, they make a substantial difference for that too. That's it. And here's the references. Thanks.
Okay. So question. Really don't everything that curious about the problem you showed about asymmetrical state in one constant. So you said two things that caught my attention. One, that possible further anomaly, and two, that it simulate on.
Yeah.
When so I guess the proof way when you apply genetic facts, does this go away? Like, is this the result of the fact that larger body masses are mammals?
Oh, so well, I'll let you finish asking your question. No.
That's the but if you control for biology, there's an empirical asymmetric phenomenon.
In specific bird and mammal. So trying to work out if I understand what you're asking. So the simulations we did were we took a mammal phylogeny. Mark Holder at University of Kansas did these simulations. He's one of the co-authors. So you start with, let's say, a mammal phylogeny. Just take that to be fixed, whatever mammal phylogeny you get. And then we're going to simulate character evolution going down that phylogeny, imagining two characters which don't have to be, like just two abstract characters. But every time there's a time passes, they evolve, and they evolve in a manner which is random variables, but with ATAs between their evolution. And all we showed was that when you set it up like that, you end up with ATAs you end up with your two characters are still correlated at the bottom, and they're correlated with ATAs at the bottom. So the evolutionary characters have to be like the evolutionary increments have to be correlated with each other and with ATAs. And I don't know, are you asking like if we got rid of the correlation between the evolutionary increments? That
What I missed that.
yeah, right.
Yeah, so.
All right. This, yeah, it's two separate. This is mammals and this is birds. Right. Like turn it into a multivariate analysis. I think you can. So I'm going to mumble some stuff, but we haven't done much of that. But I think there could be a lot of potential. So mostly when we've had data from lots of species or lots of locations, we do it pairwise. That's just better. We'll do all the pairwise comparisons, and then we'll summarize those results. Just because, like, you know, you have to do something when you start. And but then I'll say, like the ATA ideas started, the first paper that we wrote about this was something like the importance of copulas and their importance for ecology. And the idea, the statistical idea that ATAs that emerged in our thinking from that idea is this idea of a copula, which is a more general and complicated mathematical object. And let me say just a few words about what the copula is, because then it relates to me trying to answer your question. So if you take if you take a bivariate random variable, let's call it x comma y, there's the marginal distributions, like just the distribution of x considered by itself and the distribution of y considered by itself. And then there's the joint, like the joint information, how they're related to each other. And the whole bivariate distribution, there's a theorem by Sklar, which says like the entire information content of the joint distribution of the two things is completely comprised of those two sets of information, the marginal distributions and then the information about how they're related, which is the copula. That copula is like a sweeping aside, like exactly how you work that out, but the copula is the rest of the information aside from the marginal distributions in the joint distribution. And that's a complicated mathematical object. It's like got way more information than the ATAs. So like if the correlation coefficient of two random variables is one number that we use to characterize a super complicated mathematical object like this. We're taking basically one dimension from an infinite dimensional object. And when we take correlation in ATAs, we're taking two dimensions. So there's like vastly more information in the copula. And what we just found is we started out by asking like, what's the importance of copulas in ecology? And we kept coming back to ATAs kept being really important. So then we just started talking about the importance of ATAs in ecology. But probably you could, if you got lots of data, you're gonna need a lot of data, you can study copula structure in ecology and evolution and try to understand more. You need more data because when you get into more of those axes of information, you just need to get enough statistical power to assess it. You're going to need that. Now, let me come back to your question. That's just still, I've only been talking about bivariate distributions like two species or two whatever. But there's a whole theory of multi-dimensional copulas, and that could be applied. So there's a whole statistical theory. People have worked out tons of stuff. So if you've got really long multivariate data set or a really big multivariate data set, there could be some interesting stuff there. So if you're talking about a relationship between the environment and some population thing, instead of doing that, it goes up and comes back down. We haven't done that in the context of ATAs, but that could be interesting because then, like, depending on how your environmental variable is distributed, if it spans that whole hump, then that means both the right tail and the left tail of the environment are going to contribute to the left tail of the populations, because like it's bad for the populations when the environment is really low. It's also bad when it's really high. So there's like this folding process where like both tails are going to go into one tail, and the middle of the distribution is going to contribute to the right tail of the populations. And I haven't thought about it beyond like that mumbling that I just did, but it seems like it could be interesting. And maybe a good easy way to start would be to do some simulations and see what you got.
But the environmental thing, that kind of logic behind the mechanism underlying the APAs. So I'm assuming like you just said like you have some environmental pressure that affects some population. I'm assuming it's something like just the growth rate of the population.
Yeah.
Traffic parameter.
Yeah, I mean, I guess it could, if you had like an annual species or something, then it could be population, like if you census it, spring rainfall contributes to abundance, and it could be like that. But yeah, generally it should be growth rate.
Right. So I was wondering if you had any intuition about like, what if there's some coupling, some tight coupling between not the intrinsic growth rate of a population and the environmental fiber, that's some other, like some interaction parameter or like a resource parameter, some more complicated downstream, or just a different demographic. If you have anything about what times that would produce.
I don't, and it sounds like another interesting thing that you could try to figure it out, right? I have no idea. Like if you've got a complex model with interacting stuff and who knows what's in there, and you're driving it with some bivariate random variable which is symmetrically distributed versus has some kind of ATAs, what difference does it make in the output? I don't know. And so far we've just done like simple things like this. And that's, by the way, your question, these are two sides of related questions, right? Like you're taking a model that has a different model than what I considered and driving it with environmental variables. And you're taking the next step and saying like, let's make it more complicated and have interactions. It could be cool. And I don't know the answer. I wouldn't be surprised if there was something there.
The really extreme events of APA, different kind of things about like, oh yeah, this job, for really extreme events, often what we end up doing is search model is kind of occurrence, right? And so can you talk a little bit about when we choose to release that APA framework for an extreme event that's versus like we would say, okay, you have some sort of distribution and then it's a binary choice, are we in total zone versus extreme in that model? Right.
I don't know, but I guess that you might do something like that when it sort of doesn't matter. Like Lauren likes to make fun of me that I always use the example of a nuclear bomb. Like a nuclear bomb is a very synchronizing event because it just wipes everything out at the same time. So there's no difference between like, you know, if I drop a nuclear bomb that's got 1 megaton versus 50 megatons on your plots, it makes no difference at all because the whole thing is just wiped out. So in that case, like you might as well model it with a binary because there's no difference in the tails. Like the effect, it's like a sigmoid effect where you're way over in this tail and there's just doesn't matter. So it can be binary. So if it's a case where there's different gradations of how bad it is, then that's the case where I think you want to think about like how much more correlated it is as it gets further out in the tails. And I think like space should come into it too, because it should be the case that like more extreme weather events are also bigger spatially. Like you're not just going to have if you have a big thunderstorm, it's not just like over in 2 minutes, but bigger. It's over in more time. It lasts longer and it's bigger. Like things don't just get more intense. They also tend to spread out spatial and temporally. So I think what that should mean is that like when space plays a role, if you've got two different locations which are a given distance apart, and you imagine like random weather patterns going across, the big ones are big meaning more intense, but also spatially more extensive, are more likely because they're spatially more extensive to hit both your sites. And so that means automatically that'll be built-in ATAs because like the big ones are present in both sites, but the small ones might just hit one site. And big and small means both intense and spatially extensive. Right.
Thank you, Dan. Also, time